Good morning. Just trying to figure out how this works. Good morning, everyone, and thanks to Adam and the whole group for this great meeting. Thanks for inviting me, and to the previous speakers who I think really nicely set up this final talk. So I'm Beth Kirkpatrick from the University of Vermont, and I'm going to talk about the National Institutes of Health, the NIH, human challenge models. And as Steve mentioned before, our motivation for doing these are the same. I took out my picture of Charles Craig from 100 years ago in the start of these models. But again, countermeasure development, understanding the natural history and the immunology of dengue, developing vaccines and drugs. So that's what I'm going to try to get to today. I'm just trying to figure out where I'm looking here. Oh, here we go. I'm going to be talking about developing these models and then using them in a variety of different ways, particularly to test the tetravalent vaccines developed by the NIH, as well as more interesting things, such as a testing of a trivalent vaccine used in endemic populations, and also eventually getting to antivirals and monoclonals. And we'll talk at the end about the critique of the model. And I think all of these models have their strengths and weaknesses. Together, they're quite complementary, but they're certainly not perfect. And I wanted to do a shout out to the team. This group is a team that's been working together for 15 to 30 years between the University of Vermont, the NIH, and Johns Hopkins. I think led at Hopkins by Anna Durbin and the NIH by Steve Whitehead. Many of you know them. And most of these products, the challenge strains and the vaccines, are all based on viruses developed at the NIH, and most of them are attenuated with a 30 nucleotide deletion in the 3' untranslated region. So, I'm struggling to see the other screen here. So this is just a summary of what this group has done, and the bodies of work that come from these groups is really astonishing. And so just as sort of a background, this group has worked on a whole body of work that has been primed to develop these vaccines, including the start of the testing of the monovalent products, multiple testing of 5 different admixtures of tetravalent products, 1 or 2 dose tetravalent vaccines in Flavi-naive versus Flavi-experienced. Most of those were yellow fever participants, so that was discussed yesterday. We've done studies in people over 50, multiple studies with human challenge models, studies in endemic settings, and as you know, since this vaccine is now purchased by multiple pharmaceutical companies, phase 3 trials and a lot of immunology is behind this. This today is going to be mostly the clinical work. I won't really touch too much on the immunology. So this group so far has done 9 human challenge models with almost 400 volunteers. So the first 2 were to develop the 2 vaccine challenge strains, not vaccine, but challenge strains, the Dengue 2 and the Dengue 3 strain. 6 of them have been to test vaccines in various and sundry combinations, and 1 has done antiviral testing, which is the Johnson & Johnson drug that was discussed by Alan a minute ago. There'll be another talk this afternoon with more data on that, so if I run out of time, we're going to skip that. So challenge models, this is a slide that's from a general challenge model talk. They really kind of come in 2 flavors. You have to really focus on your primary endpoint, your case definition, because you really need a safe, reproducible, and high attack rate, so you can use a low number of people and still get an efficacy read. So you have to pick your primary endpoint. And in challenge models, you usually either pick a clinical disease endpoint or you pick an infection endpoint. The models I'm going to talk about are really infection models. The primary endpoint is viremia. Secondary endpoints are often rash or some cytopenias, but we don't go for dengue fever. And that's a clear difference between the model that Steve discussed and these models, strengths and weaknesses for both. So this is, just so you know, other models that use an infection model include malaria, pneumococcus. Other disease models include many of the enterics. So just to keep that in the back of the mind as I move forward here. So to develop the Dengue 2 challenge strain, all of these were initially thought to be virus vaccine candidates, and they have the 30' nucleotide deletion. And this strain was originally from the Kingdom of Tonga from an outbreak in 1974. We initially tested this vaccine as a challenge agent, and I think we totally lucked out, because a lot of times you have to do dose finding, but we used 10 to the 3 data that was initially from the use of this strain in non-human primates. And we tested 14 volunteers, 4 of which got placebo and 10 of whom were challenged. And what we do is similar to other challenge strain testing. These are outpatient studies. They're back in the clinic every other day to see a clinician, to get safety labs, to get viremia data, serology, and blood for exploratory immunology. And then we look for viremia as a primary endpoint, rash as a secondary endpoint, and other safety features. And so this in the circle is this trial, and we were lucky enough to see that all of the challenge volunteers did have a dengue 2 viremia. 80% have a mild to moderate rash, and a few have very short-lived mild cytopenias, usually neutropenia and slight thrombocytopenia that then rebound immediately. And the placebos, of course, get none of this. So the viremia for these is actually very low. So this is now PFU per log PFU, and only 2.5 for the dengue 2 challenge models compared to the NIH vaccine, which is always viremic as well for every serotype, which is about a half a log. So it's a little, this is a earlier viremia. People get viremia about day 4.5, lasts about 5-6 days. So a very short and low viremia, but consistent and reproducible. The rash on the right is the NIH tetravalent dengue vaccine rash, which is a minimal, highly asymptomatic, not noticeable by the volunteer. These rashes from the challenge strains are a little more erythrodermic. Still mild, they go away quickly, but clearly noticeable to the volunteers. So the dengue 3 challenge strain, the production of this challenge was essentially identical. This is a strain called Sleeman 78. It was designed exactly the same to be tested in 14 volunteers with the same 10 to the 3 dose. And the only difference here, the clinical outcomes are the same, essentially. The only difference here is the peak titer is actually a little lower than dengue 3. It's quite low, 1.8 logs mean. And the percent with rash is about the same, 80%. And none of these volunteers get fever. No one has elevated liver function tests. Again, it's not a disease model, it's really an infection model. Interesting, this rash consistently, because we've done a lot of these dengue 3 challenge strains, consistently is hotter than the dengue 2 rash, even though the viremia is lower. We can't explain that. And this is what we get for the curves for both the neutralizing antibodies for these strains and the viremia. Again, we're usually just seeing the predominantly dengue 3 antibodies with some minor cross-reactivity from the other serotypes and a short-lived viremia. So these two models, they give viremia in all the volunteers. It's very easily measured. It's quite low. It's consistent. They all seroconvert. We get rash in about 80%, some transient cytopenias, and really mild symptoms. So then we have to use these models. And what I'm going to talk about first is how we've been using them to test the tetravalent vaccines. These clinical studies are essentially identical for all of them. So this will go pretty quick. But what we usually do is we take US-based naive volunteers that have been tested for all flaviviruses and are naive. And they get either vaccine or placebo. They're all challenged 6 months later. We've tested both the TV003 and the TV005 vaccines against both dengue 2 and dengue 3 challenge. And since the attack rate is so high, you can use a very low number of people to get an efficacy read. And then I'm going to talk about our studies with these two challenge models with a trivalent vaccine. I'm not going to talk about this vaccine efficacy at 1 month study, but to suffice it to say it's great. And then I'm going to briefly talk about study in Bangladesh and then the Johnson & Johnson product. These studies are all the same. So as Steve mentioned, you're going to take your volunteers, and they're going to either be vaccinated or they're going to get placebo based on a ratio you're choosing. You're going to take a defined interval to monitor them for any vaccine side effects, challenge everybody. And so at the end of your controlled observation period, you're hoping that your vaccinated volunteers are protected from the challenge, and then you're hoping your placebos are infected. But you can hope all you want, but then reality hits, right? So this is a study for our first challenge trial that was used to test the TV003 tetravalent vaccine. Now this is several years old. It was published in 2018, and it was about as clean as it could be. So all of the folks that had the vaccine did not get viremia. Everyone who was a placebo did get viremia. The peak and the peak of the viremia and the duration was the same as in the model. And then the rash was essentially as we had expected in the model too. So about 80% rash, onset about day 9, and lasts for a few days. So we then subsequently did the same thing with the TV005 vaccine, and for those of you who haven't heard the talk about the tetravalent vaccines from the NIH, these are really the same. The only difference is a 1 log higher component of dengue 2 in the TV005 vaccine. And so we have been so bad at getting our data published. We've been taking massive trials, sticking them together in a single publication. So this paper was 2 of these trials in a single publication that came out in JCI earlier this year. Study 1 was testing TV005 against dengue 2, and study 2 was testing 005 against dengue 3. They were designed exactly like the TV003 study I just referenced, and really showed essentially the same thing. So the data shows that those that had TV005 and the dengue 2 challenge had no viremia, and those who were placebo had 100% viremia. Interesting, and also as mentioned by Steve, in the TV005 dengue 3 challenge, three participants did not get viremic. And we also don't know why that is. All the QC has been done properly. We don't know why that is. So the viremia, the rash, and any of the cytopenias were very consistent in this. And again, as a bar to weed out vaccines, this is pretty promising, I think. So I want to move on to this study that is yet to be published, and it's a trivalent vaccine. And this is, I think, one of the more interesting studies we've done. The question here is, do you really need a tetravalent vaccine? And we've all said, oh, after you've had 3 natural dengue infections with 3 serotypes, you're probably protected against the 4th. Well, can we test that in people that then get all 3 of those exposures at the same time in a single vaccine? So we made a trivalent vaccine with dengue 1, 3, and 4. 6 months later, we challenged them with dengue 2. And again, our primary endpoint is viremia, rash is secondary. And so viremia with this dengue 2 challenge looked like this. So by culture, about 27% of the people that got the trivalent vaccine still had dengue 2 viremia. And when you run it by PCR, it's higher, but the culture, of course, gives you the best measure of live virus. So similarly, 27% of those people also had a rash. And so what we're showing here essentially is that you do need all 4 of these to consistently get, you do need all 4 serotypes in a vaccine to consistently get protection for all 4 serotypes. Now, 73% is not horrible, but would you want to design a vaccine knowing that? Probably not. But this is really, I think, very interesting data, and a lot of immunology can be done on the back end, and it helps us sort of think through, how do we think about homotypic immunity, and do we need all 4 serotypes represented? You could ask, in those trivalent vaccinees who got breakthrough, do they at least have lower viremia? And the answer is no. And so it's a complex slide, but in the circle, both the vaccinees and the placebos that had dengue 2 breakthrough had the same viremia. So those folks were not protected at all from dengue 2. We can talk more about that later if folks are interested. And this is the print data, as well as the print data for both the post-vaccination and the post-challenge. As you'd expect, after a trivalent vaccine, you don't have a lot of dengue 2 antibodies, but then after challenge, you have about a balanced immune response without a lot of boosting in the other 3, which suggests they're fully protected against those other 3, and the dengue 2 antibodies are balancing out. I'm going to just quickly mention an ongoing study using this challenge model, the dengue 2 challenge model in Bangladesh. There's a lot of test pressure with these challenge models to make them look like a field study of phase 3 dengue vaccine trial. Probably never going to really happen because there's too many variables in the field, but we wanted to move this challenge into an endemic population to see if we can understand these variables better. And so we've had a long collaboration with the ICDDRB in Bangladesh. And so what we did is we took naive Bangladeshi adults and we gave them vaccine or placebo, and then we challenged them later with dengue 2 6 months later, or 12 months later, or 24 months later. So we're adding in the ability to look at the durability of this vaccine as well. And at the same time, just to make things more interesting, there was a major dengue 2 outbreak in Bangladesh in 2023. And this is just a slide to show that in Bangladesh, the situation is really quite terrifying. They had an outbreak with 3 times as large as a previous largest outbreak in 2019, with 80,000 cases a month and many, many deaths. So this really has swamped their health systems, and they're really desperate for a vaccine. So all this to say, this is probably not going to change our challenge trial, because we're only challenging people that stay naive. But essentially, it eliminated a lot of our placebos, because many of them got dengue and then had to be taken out of the trial. So this is going to turn into a big natural history experiment. But that data will be out this fall, and it's the first time a dengue challenge strain has been used in an endemic country. Okay, and I'm going to quickly close with this Johnson & Johnson antiviral, which you'll hear about more later on today. This is the first time we've used the challenge model to test an antiviral, and J&J 1802 is a NS3, NS4B antiviral, which is a pan-serotype oral small molecule, and it's been through this study. This picture is actually a panel of different clinical isolates and virocells, and this is the EC50 of the product. But in addition to the virocells, it's been through animal testing, as was mentioned by Alan, and also a phase 1 trial of just the product. So we move this into the dengue 3 challenge model, and this is a bit of a hard slide to read, but essentially healthy participants received a high, medium, or low dose. If you focus on the upper part, the high dose, 11 volunteers with a high dose and 6 placebos. What they do is they take the antiviral 5 days before challenge and 21 days after challenge. And they receive daily dosing for these 26 days, and then there's intensive monitoring, including viremia, safety measures, and PK testing. And this slide is from Anna Durbin. So I'm just going to show this one slide. The primary endpoint was actually area under the curve for viremia for the first 29 days, which is in the right side. And this is the high dose only, which is, I think, where the money looks like it's going to be. And there's a clear difference between how much viremia you had if you received the prophylactic antiviral versus the placebos. And I think if you focus on the high dose on the bottom right, you can see that 6 of the 10 volunteers got no viremia at all. So those that had received the product, the antiviral, 6 of them had no viremia. The 4 that did have a viremia was a little bit shorter, a little bit lower. And the lower doses and the medium doses were not as obviously beneficial. And obviously this afternoon there'll be more information on this, but it's a very exciting antiviral, I think, and really a great use of this model. All right, so I'm going to just end there by talking about the critiques of this model, which are many, and I think no sort of talk on models can be given without this guy E.B. Box that says all models are wrong, but some of them are useful. And the critiques are that the viremia is too low to reflect natural disease, and the assumption here is that's okay, that we really are focused on the infection model. Most dengue is still asymptomatic, so we find this actually still to be quite useful. We also have been critiqued by only using a 6-month challenge period, and I think we're starting to get around that, particularly with the Bangladesh study. This does not match some of the data coming out in phase 3 trials completely, and there's so many variables involved there. I guess I'm not surprised. The dengue 3 challenge strain is the same strain as in the vaccine, and that's just a fact. We're missing models for dengue 1 and 3, and the use and licensure pathways, as Kirk has mentioned, we're still working out. And the biggest critique is that we take too long to publish our data. So I'm gonna say the next things we're gonna be doing so the summary is that these challenge models are useful, very labor intensive. They're all using GMP lots under INDs with human volunteers, really labor intensive. They're predictable, they have low-level viremia, short-lived cytopenias, and a moderate to mild rash. They can't replicate a phase 3 trial. And next up for what we're planning to do is a dengue 3 challenge model in Bangladesh. I think for this vaccine, that efficacy for dengue 3 in endemic populations has not been shown, and I would love to do that. We also have more work on antivirals and monoclonals coming up in the US groups. And then with any luck, we'll get a dengue 1 and 4 strain constructs, and there's a lot of immunology on the back end that we can present at other times. So I'm going to stop there, and thank you very much.